This is my first video on muscle, and it focuses on muscle proteins. And as you can see from this fantastic statue to the right, you can see as the skin's removed, you can see muscles all over this statue. This was done by Damien Hirst. It's a rather controversial piece because it was named the Virgin Mother. I saw it in London in 2006. And it's 35 feet tall and 13.5 tons. And the pose is after Degas' little dancer pose. Kind of interesting. Learning objectives. When you finish this, you should be able to understand the structure and function of striated muscle proteins. Explain the roles of tropomyosin and troponins, TNT, TNC, and TNI. Describe the roles of calcium and ATP in muscle contraction. Describe the muscle contraction mechanism. Name the major storage form of energy in muscle. Describe smooth muscle contraction and tell how it differs from striated muscles. Describe the roles of nitric oxide, cyclic GMP, and phosphodiesterase in smooth muscle. And here is a picture of muscle. And so here's a muscle in your biceps. And the muscles have sarcomeres within them. There are about 100,000 sarcomeres per every muscle cell. The muscle cells have a lot of mitochondria because they need to produce a lot of ATP. And I won't go into all these details, but you'll see these again in cell and tissue and in physiology. But primarily going to focus on the muscle proteins. You don't really need to remember what the different bands are for me, but for biochemistry, you'll have to learn that later because that's what you'll see in cell and tissue. So here's a sarcomere, and it has heavy bands, dark bands in the middle, and light bands on the ends, and then you have a little connection line, the thin filaments. The dark bands are due to the myosin heads. The myosin is the orange one in the middle here, and it's the big protein. You see a lot of that. And then the thin filaments are the actin filaments. So actin and tropomyosin wrapped around that, and then you have the TNC and TNT and TNI proteins on there. We'll talk about those in a minute. And you can see as in this animation, as the sarcomere contracts, shortens the muscle. That's how you get muscle contraction. It's a molecular motor, and it uses ATP to do this. And so what happens is the heads pull out and pull the actin in, and so together shorten that actin, shorten the filament, and you get muscle contraction. So the actin, G-actin is monomers of actin molecules, and they bind ATP and magnesium. And when they do, then they'll form polymers, and that's called F-Actin. The F-Actin will form these long polymers in red, and it's wrapped around in black with the tropomyosin here, tropomyosin. And then you have a troponin complex. You have the I component, the C component, and T component, and those are regulatory components that help this thing work. And so the actin monomers get polymerized into F-Actin filaments. So you have F-Actin is a fibrous actin, G-actin is the monomers of that, and it forms a two-stranded helix. Tropomyosin wraps around it, wraps around the F-Actin, and troponin has three proteins, TNT, tropomyosin binding, TNC, calcium binding, and TNI, the regulatory subunit. And you can see what happens is when when calcium binds to the TNC, the calcium binding module, then you get movement, and then that movement allows the calcium binding to TNC exposes the myosin binding site on the actin. So normally that binding site on the actin where the myosin head could bind is covered up, but when the calcium binds, and that's caused by nerve firings, and opens up the sarcoplasmic reticulum and lets calcium into the muscle, and then it will contract. So here's the a little bigger picture, you can see you get this helical helix probably here. That's not an alpha helix, but a long helix of myosin, and then you get ATP binding to the two heads on the myosin heavy chains. So myosin is 55% of muscle protein. There are 6 chains, 2 heavy chains and 4 light chains at the two of 2 different types. Different muscle types have different light chains. There are 294 myosin molecules per thick filament, and so you have 588 ATPase sites. So these heads will hydrolyze ATP, and that's how the mechanism works, and we'll talk about that in the middle. This is a nice picture by Robert Finkbeiner that I found several years ago, and it shows it was drawn in real artistic type form, showing you the various components of the myosin. And then here's the muscle contraction mechanism. So the contraction mechanism starts out with, you can see that F-Actin filaments in blue, calcium binding here in red, tropomyosin sort of an orange here in the back, and here's the myosin head with that has ADP and phosphate attached to it. And that head will then attach to the calcium. So calcium binding to TNC, and then the myosin head that has ADP and an inorganic phosphate bound to it will bind to the actin. The next thing that happens is you get what's cross-bridged. This binding is called cross-bridging. So once this is bound, it's cross-bridged from the myosin to the actin, and so those are stuck together. Then you get the power stroke. In the power stroke, ADP and inorganic phosphate are released. And so once that happens, then you get a conformational change in the myosin head that shortens the whole sarcomere, and you get the muscle contracts. Then ATP rebinds to the myosin head. You get relaxation, and so it can relax out, and then it can that gets hydrolyzed, and then you can start this whole process over again. It can rebind to the actin, and then shorten and then bang, and then release and shorten it each time. So you get multiples of these occurring, and so you get ATP hydrolysis in the Cox state when you get back to the Cox state. All right. So here's a little video from the University of San Diego. As you can see it binding, you see the calcium binding to the change in the structure of the troponin complex, and when that does, then the myosin head can bind and ATP gets split and all this sort of stuff. Great. It's a nice video. So again, you can see the shortening of the sarcomere. Muscle energy sources. Resting muscle uses glucose, amino acids, and fatty acids. High blood glucose stored as muscle glycogen. And low blood glucose, when you don't have a high level of blood glucose, the muscle uptake falls, so glucose is shifted to the brain and red blood cells. So if you have a low blood glucose, the muscle uptake falls, so you can shift your glucose to the brain because you need that mainly there and in red blood cells. Isoleucine and valine yield 6-succinyl-CoA for the mitochondria. So in their metabolism, we'll talk more about that in amino acid metabolism, and they result in making acetyl-CoA from fatty acids, and so the fatty acids can provide a source of energy for muscle. Fatty acids, the main energy source in starvation for muscle. Myoglobin in muscle stores O2 because you're going to need a lot of O2 to do oxidative phosphorylation that produces more ATP. So muscle energy sources again, intense exercise, you're doing mainly anaerobic and intense exercise, you're anaerobic, such as sprints and weightlifting. ATP is produced by glycolysis using glucose and muscle glycogen. The anaerobic metabolism, you're producing lactic acid, you have a lower pH, and you can have pain from that, from all the muscle work you do, and that lactic acid building up, and the lack of having oxygen to the tissues. High enough O2 for oxidative, not enough O2 for oxidative phosphorylation. That's why lactic acid builds up. Moderate to long-term exercise, such as aerobic exercise, walking, jogging slowly. ATP from muscle and liver glycogen is where you get your ATP, amino acids and fatty acids, and you have enough oxygen then to do oxidative phosphorylation pretty well. So here's a little schematic. You don't need to remember this, but you notice that the ATP gets used up pretty fast, and then we switch over and use creatine phosphate as an energy source. That phosphate stored in the creatine can then be used to make ATP and keep your ATP levels high. In anaerobic, we use mainly glycogen to lactate, and in aerobic, we use glycogen to carbon dioxide over time. So here's the pathway for the synthesis of creatine phosphate. It starts out with arginine and glycine in the kidney, and then that goes to form ornithine and guanidinoacetate, GAA, guanidinoacetate. And the guanidinoacetate then gets transferred to the liver through the blood. Once it gets to the liver, you're having S-adenosylmethionine provides a methyl group from methionine to S-adenosylmethionine provides that methyl group, and then you have an enzyme called guanidine acetate methyltransferase, and that puts that methyl group on the creatine, on the guanidinoacetate to make creatine. And so here's creatine, and the byproduct of SAM, when SAM gives up its methyl group, it forms homocysteine, and the homocysteine can pick up a new methyl group from serine, and that can form cysteine and glutathione here. And you can also recycle this homocysteine back up here with methionine synthetase, which requires B9, B12 cyanocobalamin. We've talked about those before, to form S-adenosylmethionine again. So the creatine goes into the body, and this is the creatine molecule here. You see it looks just like glycine here with a guanidinium group here from the arginine on it, and then the methyl group that came from SAM. And so here's the way it gets phosphorylated. ATP will phosphorylate it. When you have plenty of ATP, you store that ATP over at creatine phosphate, so you put a high energy phosphate group on here, and then when it breaks apart, it takes goes back to form ATP, and then you get creatine. Again, eventually creatine can be broken down into creatinine, and the creatinine can be observed in the urine, and it's a measure of muscle mass and of kidney function. Creatine supplements increase creatinine and can cause errors in kidney function tests. So if your patient's taking creatine supplements, and a lot of bodybuilders do that because they want as much creatine phosphate as possible for the energy, then measuring glomerular filtrate rates doesn't work quite as well. So, all right, you need to know that. So here's the guanidinoacetate